So this is published in the journal PLOS Biology and is entitled Food Hardness Preference. I did read that correctly. Food hardness preference. So what is the texture of food? That's the takeaway message here. So in this case, Spartacus, it was for rotten and ripe, rotten versus ripe. And the reason it was being asked was, we know insects, particular fruit fly larvae, right, have preferences for certain types of food, right? Are there textural preferences? So we know their smell goes into the flavor pile. We know that taste goes into the flavor of what kind of food they're going towards pile. But what about texture, right? And found neurons. They're working in fruit fly larvae. They found neurons in the mouth of the fruit fly larva that allows them to detect the actual food texture. So they found this tasting, texture detecting organ that has a bunch of texture sensing mechanoreceptors. So mechanoreceptors are neurons that detect structure, or they detect what kind of surface that you're walking on. So it turns out these were also derived from genes, or like similar to genes in terms of how they're structured, that detect pain and feeling, except this is linked directly towards taste. So a lot of past research in fruit fly larvae, and we'll take a look at some of the examples, focused on things like if it tasted sweet or salty or basic or acidic, and not really texture dependent, right? So here's an example that y'all are going into in chat of a food item like peanut butter, but there might be peanut butters that you don't have a preference for because of the texture, right? So the crunchy versus creamy peanut butter argument, right? You might like the taste of peanut butter, but you might hate the texture of really crunchy peanut butter. Ones that are governing your texture preferences. It's not actually your fault. You have neurons that are like, oh, this is what you like and this is what you don't like. Mushy brown, yeah. So Coffee Rocks, that's exactly what this paper is getting at. So one thing that I like to do, Coffee, is to look at the abstract first, right? So the abstract, for those of you who haven't been here when we've kind of broken down this different regions and like structure of a paper, this is supposed to be like their summary. This is where they're like giving us, here's the problem statement, so here's the issue that we have, and here's what we did, and here's what we found, and here's why everything is so cool, right? So food presents in multisensory experience. Visual, taste, olfactory cues are important, right? You gotta see it, you gotta taste it, you gotta smell it. The argument is food multimodal in terms of detection. And what they even pointed out, texture is largely unexplored, despite providing key information about the state of the food, such as hardiness, liquidity, so like how broken down it is, and how granular it is. So food perception achieved by specialized sensory neurons, which themselves have defined by the receptor genes they express, meaning that there are particular neurons that are responsible for detecting certain things, right? There are neurons that detect taste, that there are neurons presumably in your mouth that contribute to that preference, which makes sense, right? Like, why else would you like crunchy food over soft food if there wasn't some neuron detecting that texture, right? And again, why aren't we studying in humans? It's just easier right now to study it in fruit flies. You can move up into higher level organisms. There are so many genetic tools in this animal that you can actually figure it out, right? And so they're saying, chocolate taste system, there's gustatory neurons that respond to this hedonic valence, so that's like acidic or basic, and then olfactory, so smell cues. But here, we identify that larvae ingest and navigate towards specific food substrate hardness and probe the role of these neurons in this behavior. So a new kind of neuron they identified. They developed genetic tools that target specifically the gustatory organs and show that these organs are major contributors for the evaluation of food hardness, ingestion, decision making. You train them, be not sensitive. You can train them, chat, not necessarily for sensitivity, but preferences. The sensitivity argument is linked back to what are called memory extinction genes. So these neurons, usually like any other neurons, they undergo neuronal pruning, and that pruning is involved in you having a preference for something. If you were to turn on memory extinction genes in those neurons, the hypothesis would be is that you can prevent the neuronal pruning, and you could have it be like a blank slate where it's less of a preference. However, that would take a couple of rabbit holes of experiments to be able to identify, but it could be possible to do. And so one cool thing that they did here is they ablated the gustatory organs. So this is again why I'm like, I think y'all, based on what we chatted about this week, y'all would enjoy this. They identify this set of neurons that's important. They identify this gustatory sensing center in the head of the fruit fly larva, and then they just get rid of it. So ablation doesn't mean that they like chop it out. It's like we just genetically just killed that organ. Now let's see what happens. And it turns out if you just kill that organ, their navigation and ingestion preference very much varied. Now they liked hard substances all of a sudden. They finally found that some neurons are variable and they have physiological responses to both mechanical and other kinds of stimulation. So they're detecting both smell and texture at the same time. And then individual neurons house independent mechanisms for multiple sensory modalities. And so it's challenging the idea that sensory neurons only get sense, but they can detect other things at the same time. So depending on how you're asking your question, they never would have seen this, because you then have to ask your question of like, okay, are these neurons involved in sensory detection, both on, odorants and touch? All of those are kind of coming into play here. So again, just some big hitting highlights from the paper that I thought we can get into, and then we'll kind of go into some of the background and we'll go into the figures as well. Why the experiments here are so, I think, clean is because they have a clear behavior, right? The larvae either they don't like the hard food, they don't like the really soft food, they like it just right. And if you get rid of these neurons, now all of a sudden they prefer anything equally. And so the idea is that, yeah, they might be able to chew on any of them, but as Sparky points out, they might not be able to digest any of these. There might be other things that they could get sick from, right? I'm guessing that some termite nymphs don't have these neurons. So actually, Jay, two answers. One is the nervous system of termites is severely understudied. Actually, three things. Number two, this was done in fruit fly larvae, not the adults. And termites are complicated in that they have a nymph stage, from larva nymph to adult. And it was the nymph eating from the butt of the adult. And then the third difference is like there's just no trophallaxis in fruit flies. So even the inherent phenomena that we would have seen just like don't happen to them. But I would, if I had to guess, they might have these neurons. God, I can't believe I'm going to say this. But Jay, they might have neurons that make them prefer the taste of butt or the texture of butt. Jay, we're going to get in trouble. We are 100% going to get in trouble, Jay. But said hypothesis makes sense, right? I know Falcon 2 is like, listen, say to me here. So Jay, if the argument is that there are neurons that detect texture, and that texture detection is important for the animal to know what to eat, that would suggest that the termite nymphs might have neurons that make them to prefer the texture of butt. So let's go ahead and get into the meat and potatoes of the paper. There's actually quite a cutie patootie figure here. But so yeah, what I really like about this paper, folks, is that they actually have a really straightforward explanation of the experimental idea. Like, what kind of fruit will the larva like? Will it be full rotten, or will it be this intermediate state? And then they check multiple different kinds of fruit, and you can see that there are like set preferences during when they like something. So like the apple's decay day goes down at day 5. The worst offender, right, is the banana that they prefer like on day 1, and then it just plummets down. If you touch the piece of fruit, is it soft or is it hard fruit? So that's the compression index that they're measuring here across different days, and just figuring out like what preference there is for the animal. And they find, yes, the animals have a certain preference across certain days of certain kinds of decay. So that's step 1, right? So step 1 is they had the hypothesis of, we think there's neurons that control something like this. First, we need to demonstrate and replicate that you can do this in the lab setting. So demonstrating that there's a certain level of softness of fruit that they don't like, a certain level of hardness of fruit that they don't like, and something in between that they do. Next, here you go, Jay Spites. So first thing first, they needed to figure out what cells they needed to be interested in. So they did a process called single cell RNA sequencing. Okay, so what they wanted to do was identify what cells in the brain of the larva had a gene called orco. But say you're trying to identify neurons that have a ability to detect odor, and those are neurons that have the gene orco that are on. And so what you do with single cell sequencing is you sequence every single cell. So just like it sounds like. And what you can do is you can cluster the results. And so here they're clustering the results based on, in red, what genes or what cells have orco. So everything here is a cell in the brain. So imagine this really beautiful technique where you can sequence cells in every brain, or every cell in the brain, and figuring out what's on and off in every cell. And I think that's a really, really cool technique, and that also gets you a candidate list of what to look at. We know which cells have peanut butter and which have orca in them. Well, so Charlie's Dragons, here's the thing. This technology is not perfect. And by that I mean it's getting better and better, but a really big limiting factor was how many genes it could detect. So if there were small changes, no, Charlie's Dragons, we're going to follow this up. If there were small changes in gene expression between cells, that wouldn't be able to be detected. You can see the larger differences, but you couldn't see those really small differences between cells. And so that's an important thing to note, is that the technology is still imperfect. So at the time, like 5-6 years ago, you can get 300 genes that you can say with confidence are different between two different cells when doing single cell sequencing. Now you can get even more. I think you can get like near 1,000. And the reason is because you're trying to measure the RNA of a particular gene in just one cell. If you do it in the whole brain, there's a lot of tissue, right? There's a lot of RNA that you can measure. But doing it in a single cell, your starting material is really, really low. And that's what makes it difficult to check. And so if there were peanut butter or there were orcas in there at really low levels, we wouldn't be able to see it. Tech's getting better and better, though. If it was at high level, then you could see the peanut butter cells and you could see the orca cells as well. Hopefully that makes sense. So that's a nice little bit of immunohistochemistry for you, Jay. Also, they were trying to replicate the experiment that they did earlier in this set. So remember, we saw they had figures where there was firm fruit and rotting fruit, and they were trying to test where the larvae will prefer. Now it's a little bit more controlled. So instead of having regular like fruit and rotting fruit, now they changed agar concentrations to make agar either firm or soft with different types of foods to see where the larvae end up going. And so it's just a little bit of experimental replication. You take out the variance of what's going on with just fruit, like random fruit that you're getting, you're able to better answer the question. So I like these kinds of papers, I Wombat, because they go from observation down to the molecular level and then back to the observation. So like here, this is another antibody stain. This is an example of a really, really good antibody working. This is an example of a good antibody working. So this first panel here, the dots here are single cell sequencing data. So every dot here is a neuron in the brain. And the red is trying to quantify which one of those neurons turns on a gene. So in this first one, which of those dots or neurons has the NAN gene on? And it says there are 2. So then what they did is they looked in the fly brain. They had an antibody stain for NAN, and then in green, and then a stain for all the neurons in purple. And this is really, really cool, right? There's 2 neurons that are glowing green. Like, how cool is that? So the single cell data that you get from the sequencing matches what you see with your eyes. Now, if you didn't see this with your eyes, it could be that the antibody was garbage, or the RNA sequencing data was garbage, right? One or the other. But they're in agreement, so it's a nice way of seeing that, yes, this is like solidifying the answer that you observe one way and you can observe it a second way. Like, I get this in principle. You sequence cells and saw what differences in the RNA exist, right? And you filter that list and you find that 2 cells express this gene NAN. Like, I get that, but then it's, I think, nicer to have this, like, all right, let's get that visual validation as well. And again, it is dependent on having a good antibody. So this LV that you see here in purple, what's really cool about that is you and I, Jay, could order that for $25, that antibody. So same principle for this one, y'all. This is looking at a gene NOMP-C throughout here. A couple of cells. You see that in the image. And then here, this is a gene called pain. What you'll notice here, there's these streaks in both. Right? It gets a little bit streaky. That's because it's the body of the neuron. So the dot here is just marking the neuropillar, like that, the center point, the center of the axon, like the cell body, sorry, not the axon and the dendrites. But then the one next to it, see, it's also stripy. That's just, again, the body of the neuron going down. But you've got a couple of cells. Yeah, so here they're marking, again, this pain gene, and that turns out to be the important one for this paper. Where here, they actually used machine learning software to figuring out what cells have the pain and the H2B. So so here you're actually doing there's this G camp here. This is actually the experiment that Jay, you were wanting to see. So this is a calcium sensor. You take the calcium sensor and you put it in the neuron that you're interested in. And now you provide that neuron the stimulus that you're working on, and you see if it turns on. The reason the calcium sensor works is because in order to have a neuron fire, you need calcium firing. You need a calcium part, like an influx. And that sensor will tell you if there's a change in calcium in the form of glowing light. And so it's cool that like this little machine learned map, you can actually figure out what component is glowing and therefore what component is important and matters. And so here you can see like the CM1 neuron is the thing that really, really fires rapidly to sucrose, versus these other neurons don't have that level of firing. You're able to identify what neurons are important. Hopefully that makes sense and y'all find it cool. And then as we go forth, I guess this was the one with the mutation as well. Find the mutant data real quick. Here we go. I can't remember if they have a figure. Well, this is good enough. This is good enough. So this figure here, we scroll on in, take a look at this panel, right? If the argument is that they have figured out the neuron that is involved in the behavior, and they're again testing their food preference. They're testing their preference for rough texture food versus soft food. Here, there's no difference. Everything's flatlined. There is no preference when you turn off that neuron. And what's cool is, so as Locke just points out, look at the letters at the bottom. Those are crazy. They tested this in multiple different ways. So this is the straight up mutation. This is when you RNAi or kill that neuron with this construct, this GR21A. You can RNAi it with a different thing, and you can RNAi it with a different thing. So GR means grim, the cell death protein. You can so you're turning off these genes, and you're getting the same results every time, meaning that they replicated in 4 different ways how these neurons are being turned off, and that makes it so they no longer have that preference. Does that make sense? These are references, not necessarily DNA structures. Lockjaws, and maybe we're saying the same thing, but structures to me represents like the 3D organization of the DNA. This is more those letters and numbers that represent a gene. And what you're doing is that gene is turned on in a particular neuron, and so you're inactivating that neuron. And then the question is, okay, if those neurons are inactivated now, what does that mean for the behavior? Does the behavior have the exact same outcome? If I turn off that gene and the behavior still works just fine, then maybe that gene isn't important. But if I turn off that gene and that neuron and the behavior totally goes away, then maybe it is important. So here we've eliminated the behavior, therefore it is important. Without the mutations, they have a sweet spot plane shifter. So they tested in the paper, it actually tested a couple of different fruit. I thought it was quite a clever way that they went about it. So they tested apple, banana, pear, and pineapple. And they tested at different decay states. So like really, really hard, like not yet decaying, to like moosh, and tested the preferences. They then replicated that in a petri dish with agar and food in it. So agar is like what gelatinizes a substance. And so you can make the agar thicker and harder, or you can make it softer. And that way it's standardized. Like you just can't compare those two. So here they got a baseline measurement with rotting fruit. They measured the tensile strength that's in there. It's like, what's the toughness and rigidity? And then they replicated that in a dish with agar or that jello. And then they use that every single time. And that is repeatable. Does that make sense? And so it turns out the texture they like without turning off those neurons is in between. Hard, like not rot, like they needs to be rotting, and not straight up moosh. It's like a Goldilocks effect to it. Flipping on and off in this animal, do the same thing in this animal they would do in another. It might. So lockjaw is definitely closely related animals. You would predict that it would. So larvae of other fruit fly species, and maybe even a little bit further out, you'd probably be like, all right, they're probably going to function something similar. Now, other organisms, right, might have more neurons, right, that control this same thing, but the basic biological principle could be there. So it's giving you foundation to be able to analyze it elsewhere. It doesn't guarantee that you'll be able to replicate it in a different organism, but it at least gives you a starting point of where to look, and then predicting that at least the phenomena could exist. And it makes sense, right? Like, I'm sure Lockjaws, you have texture preferences of different food. One of the ways by which that texture preference of different food can manifest is maybe through neurons that are in our tongue, maybe, right? Or like in your mouth that like lets you like towards certain kinds of textures. Any departure from the natural food source, how do we actually know it's the firmness of the agar and not something else when we try to standardize it? Yes. So Jay, what was cool was this here. So what they did was that the compression module of the agarose, and so seeing like how does the compression state of the fruit correspond to the compression state of the agarose percentage, so at least that they were able to make the same firmness, and they also made similar sugar concentrations to this. It's fair question, Jay, that this isn't very much like the natural food source. However, if you have the natural food source, there's also going to be other signals there, right? Like the smell is definitely going to be there. There will be a flavor to it that could be variable. All of those things together could also be skewing your data. If all those things are skewing your data, then are you getting an answer that's not biased, if that makes sense? Because it's multimodal. Yeah, exactly. Because that, not just rotting fruit, like even if fresh fruit, right? Like there's other things that you can detect. And so because of those other senses that could be stimulated, you know, it might be that I get rid of the neurons that detect tactile sense. That's fine. What if I get rid of that, but that's not the primary thing acting? Like, what if it's a series of events, Jay? Like, first thing you do is you detect via smell, and then when you go over to it, you're like, oh, it feels nice too, so I'll eat it, right? What if you get rid of then the second step, which is how does it feel? The animal can't get the textural information, but it smelled nice, so it goes and eats it anyway, right? Then does it, you know, like what are you actually getting in terms of the answer? It'll make it even more complicated. Hopefully that makes sense, but yeah, it's just an extra difficult way of interpreting the data with those extra variables. Now you're 100% right. Like if you were to put it as in nature, would it replicate? You know what would have been a nice experiment to do at the very end? Redo this first one. Redo this first experiment that shows not too hard, not too soft with rotting fruit, and see if with the getting rid of that neuron also wipes out preference. If it doesn't work, you have a ton of reasons why it might not have worked, right? Because of the senses that we're talking about. However, if it does work, then you've really nailed it down the neuron in the pathway. And you would have answered your question too, Jay, right? You would have done all the data collecting for most of the paper in a very controlled manner, and you would have brought it back at the end. That would have been maybe the way to do it. And see, that I think is a fair criticism, and one that if you were reviewing the paper, you could have pointed out and be like, you know what? It would have really nicely dovetailed if you do this extra thing. So again, just the big takeaway of this paper, and I thought it was really cool, is that they end up seeing like what detection is important, what are the neurons involved. They found that there's the peripheral taste organs. They selectively turned off the neurons in the larva's mouth, and when they turned off the neurons, they had a loss of texture detection, and tried eating food that was softer or harder than what they usually preferred. And so they found that the gene and the neurons are what's responsible for this sense. They also found that there was this extra neuron called C6 that we looked at in one of the figures that can sense both sugar and mechanical stimulation, which was this neuron down here. And the reason that was cool was because that's the first example, this one, the C6 here. So all of these neurons here, right, that we're looking at here are detecting mechanical senses, but this one also detected sugar. And so that's the first example where you get not only the neuron that detects mechanical stimulation, but it also detects taste. And that also is a new kind of way of thinking, because normally that was thought that that's not a thing. That's not how this works. You're not going to be detecting both odor or flavor and the mechanical stimulation, but yet this shows that it can happen. And that makes things much more complicated. Really cool, but it makes things more complicated. And having that example at the end of the day is really important, because it will cause you to rethink how these systems work. Because if the same neuron can taste food texture and substance, it means we probably need to rethink a lot of the questions that we've asked in the past that we thought we might already have an answer to. And so, with the power of fruit fly genetics, you can harness the hardness and softness of food and figuring out what neurons are sensing them, plus a neuron that can sense both chemical and taste. And that is the beauty of this paper. And I hope you all enjoyed it.